You're all honor students living in a dorm together, everything. I did the exact same thing. I was in an honors and research program with a group of students, and we lived in the same floor of a dorm, where I got into doing two different research projects. Part of my undergraduate degree, or the honors program, was I had to do a sophomore thesis and a senior thesis. So the sophomore thesis, I worked, like I said, on a bacterium called Bioluminescence, which is the bacteria in nature that lives inside of little worms, microscopic worms called nematodes, which infect insects, and organic farmers will spray this worm onto their crops to get rid of insects and without pesticide. And what's interesting is these little insect cadavers, once they're infected, at night you can go out and they'll glow in the dark because this bacteria actually produces a toxin, and when it grows inside of these insects, it glows. And we've actually taken the genes from this bacteria and given it to bacteria of medical importance, and I'll get into that in the next slide, to study them in vivo. The next project I did was a food safety, along the more of the food safety line. I studied a bacteria called Clostridium botulinum. Does anybody know what the significance of Clostridium botulinum is, or ever heard of it? Botulism, right, and Botox. So I worked in a lab where we studied the food safety of processed cheese, actually. So Kraft Foods is right in Madison, and my project was, people have been, for kind of the history of making like Kraft singles and all that, had always used dairy fat or butter fat in the making of the cheese. But there's been a change for cost reasons to use vegetable fats as a substitute, and nobody knew what this did for the safety and growth of Clostridium botulinum in the cheese. So my senior thesis was actually making a bunch of processed cheese, letting it sit around, and then seeing if botulism was able to grow with this new fat formulation. Needless to say, or very good thing, it didn't grow. So the formulation was safe. So the cheese that was out on the shelves was safe to eat. So the one thing that you guys may or may not know about like Kraft slices, American cheese, is that stuff could sit on the shelves, not in a fridge, for about 5 years and still be about the same thing it is when you went up and picked it at the store. So if you ever feel like there might be a food shortage or something for a long time, go stock up on processed cheese. You'll have a food source for quite a while. So that was my undergrad research. Doing the research in kind of the food safety side of microbiology, I wanted to learn more about the pathogenesis and how bacteria cause disease. And that's what brought me to Stanford, where I'm doing my Ph.D. and my thesis project is studying the extracellular physiology of Listeria monocytogenes. So extracellular, meaning that the bacteria grow outside of cells, which for this bacteria was a big finding. I'll describe why here. So here you have a mouse, and I don't know if you guys can see, but you have a little signal coming from right there. So what we did was took Listeria monocytogenes, gave it the genes from that Photorhabdus bacteria that produce light, so now this bacteria produces light. And I'll go through the physics and everything about this later, but basically we have a special camera that can sense the light, so we know that the bacteria are living in that site where the light is coming from. What this turned out to be was the gallbladder, which was very interesting because almost every mouse got this signal right here coming from the gallbladder. Listeria, before this research, was only known to live inside of cells of like the liver and the intestines and things like that. But in the gallbladder, it lives actually outside of cells. It lives in the bile that sits inside of your gallbladder. And this was a very big finding. A researcher in my lab published this research actually in Science, and I've come along to figure out what actually is going on, how the bacteria lives in this environment. So studying a little bit more about the physiology here, this is just a standard, I put bacteria into bile that I've taken out from mice. I actually take a little syringe, go and find the gallbladder, suck the bile out, put it in a tube, add some bacteria, and see how it grows. And you can see over time you get a larger population. The curve kind of comes up and then goes down. And the curve is actually the amount of light being given off from the bacteria. And it starts going down, and I can describe that later. But this is kind of the first thing about it, that we showed that bacteria are able to grow in just bile, which was a very interesting, because bile, our bodies produce bile because we think it's going, or because it helps fight off bacteria, so that a bacteria could live in the environment that produces, and something that kills bacteria was pretty impressive. My research from this has come to find out that the carbon source they use actually went down and broke bile down into all its different components, and we found out that it actually lives off of cholic acid, which is the main bile acid or bile salt in bile, which is pretty interesting. I also have done microarrays. Anybody know what microarrays are? Heard of them? So basically what you're able to do is take all the genes from a bacteria, you put them on a little chip. If you isolate the RNA from those bacteria, you wash it on this chip and a laser scans every gene, and you can see whether at one time point, whether the genes are turned on or turned off, every gene of an entire organism at one time. It's a very powerful tool. And we showed that the genes important for causing infection of this bacteria are basically turned off in bile. So we think that this environment, the gallbladder, is actually a spot where the bacteria live but don't cause infection. So every time you eat a meal, your gallbladder contracts to help you digest, and you go to the bathroom, it sheds out in the environment. We think this is a way that Listeria is spread and carried in livestock populations and things like that. And then finally, my research led me to look at how bacteria get there, either from the liver, climb up from the intestines, or through the wall of the gallbladder. And actually, we did a little mouse surgery. I'd have open up the mice, find their gallbladders, and go in with little binocular surgeon things, and find the bile ducts and tie them off with sutures. And I did 3 different groups, ones that I tied off coming from the liver, tied off coming from the intestines, or tied off both. And we looked to see how the bacteria got there, and it comes to be that the bacteria actually come down from the liver and get to the gallbladder in that way. So that's been my research. If we have more questions about that later, I'll go into a little bit more of the details later. But if you have questions, we can ask those at the end. So we'll get into now the bioluminescence. So we're going to break this lecture kind of into 2 parts. The first, we're going to go through the bioluminescence basics, like how light is produced chemically, where light comes from, the luciferases and luciferin, the enzyme and the chemical that are broken down to give light, and then a little bit about bioluminescence imaging, how we do it, how we create cells or models to study or use bioluminescent imaging to study diseases. And then finally is how we're actually able to visualize this light within the tissues. The second part after our break, we're going to go into a little bit more about how we answer biological questions with bioluminescent imaging, either be cell or disease tracking, how we can look at drug discovery processes in a more efficient manner using bioluminescent imaging, and then infectious disease like Listeria. And then we'll talk a little bit about where we think bioluminescent imaging is headed in the future, something you guys might all be interested in doing for maybe your graduate work at some point. So we'll get right into it here, part 1. And we'll start out with just the basics of kind of light production in general. So light can be produced in several ways. The first being the thing you're kind of more accustomed with, why you have lights that work, incandescent lights at your place. Heat or electricity is pumped through a filament that the electrons get excited into an excited state, and they relax, and that relaxation from an excited state gives off a photon of light. Just like in a light bulb and a candle, most of the energy produced during that is in heat, but some of it is given off as light. Another way light is given off is something called fluorescence. And has anybody ever seen a bottle of tonic water glow like that? Been in a place with black light? So if you ever order, well, you guys aren't old enough yet, but when you're at a bar, you'll have a gin and tonic someday or something like that, and they have a black light going, it's going to glow that bright blue. And what there is so tonic water, it's kind of bitter, it's sweet and bitter at the same time, and there's a chemical in it called quinine, and this is a relic of kind of British colonial times, or colonization times, across like India and the tropics, where malaria was a big deal. And quinine was a prophylactic against malaria. So people drank tonic water to prevent themselves from getting malaria. And it just so happens that this compound under a black light excites electrons, and it gives off this blue light in the form of fluorescence. So it's just an interesting real world example of how fluorescence, and we as scientists, I won't get into this, use fluorescent proteins and fluorescent molecules a lot to probe biological questions. The next big way light can be produced, and these are the most similar, is chemiluminescence. So light from a chemical reaction. What happens here is, instead of heat or energy being brought in to excite the electrons, the energy comes from either usually a breakdown of a chemical product, and the energy in those bonds during the breaking is able to give off light. And this happens in like glow sticks, and in the case of biology, in fireflies and the bacteria that produce light, the chemical breakdown gives off light, and it's just a form of chemiluminescence in a biological organism. So that's bioluminescence. This process happens in many different animals in nature. Here, hopefully you guys can see this okay. On the left, you have shrimp and krill that are naturally bioluminescent. That organism in the middle is a squid, and you can see how the light actually comes kind of right from its snout, mouth region, and that actually is called a light organ, where bacteria called Vibrio fischeri lives, colonizes this light organ and produces light for these squid. And it's thought that when these squid usually live on the ground floor, on the floor of the ocean by day, but at night they go swimming and hunting, and they go up more to the surface. And if a fish sees them, they look up, they just think that little light organ is like a star. So it's like a camouflage system, and we'll get into more of that, other kind of mechanisms of why bioluminescence is produced. You also have jellyfish and other like sea cucumbers, and I'm sorry those don't show up very well. On land, I already talked about those, are kind of a blown up picture of these bioluminescent worms called nematodes, and then you have the firefly. Those are just very few examples. So this is a whole comprehensive list of organisms that we know of that produce light, and this list could go on and on. We'll talk kind of the big ones that are out there. So dinoflagellates, I don't know if you saw my first slide, that kind of blue and red behind the title. That was actually a wave. And I'll go back into this again later, but there's a huge wave coming into the ocean, and that was light being produced by dinoflagellates, which are kind of everywhere in the ocean. Kind of neat. And then fireflies and click beetles are the most biologically relevant for us doing bioluminescence imaging. We get the genes from those two organisms. We also get them from sea pansies in a different system. So the purposes of bioluminescence. Bioluminescence can be used by organisms to, one, locate food, shine a flashlight to see around. They can attract mates or attract prey. Bioluminescence is produced to fend off predators. Can be used for camouflage. Can be used for communication. And there possibly are other reasons for it. A lot of the mechanisms of bioluminescence have yet to be elucidated. I'm going to go through each of these examples specifically, so don't worry about getting there. So here we go. So the major contributor, like I said, of bioluminescence in the ocean is these dinoflagellates. There's a microscopic image of them. The one on the left in yellow is a light microscope image, and then the one on the right is using a bioluminescence microscope that can see the light, and all those little kind of organelle-like structures are where the light is produced. And again, that's about a 25-foot wave coming in off the coast of, I believe it's Australia, taken at night, and that's light, like a big bright wave coming at you from the water from all these dinoflagellates in the sea. Communication and camouflage is another reason bioluminescence is applied. One example is krill or shrimp produce this bioluminescence, and they think this is a mechanism of communication, so the krill will stay together, they can see the light, and kind of all stay together at night, because especially with currents and things like that, it'd be easy to get dispersed. They also think it's a purpose of camouflage, kind of the same starry night type of camouflage. If a fish is down below and they're above, they look up, they just think they're going to see the night sky above them, not a bunch of krill or food. Another interesting camouflage, and this is a predator now, it's called the cookie cutter shark. So on the belly, or the ventral side of the shark, it has across its entire belly, all but this little black circle, is photophores. They're called light-producing cells or organs. And if you imagine during the day, this shark's up by the surface, and prey are down below, and they look up and they see a small dark spot. They just think it's another small fish. But in this case, this shark now has tricked them. They'll get closer to it, and then it'll attack. It uses this as like a camouflage so that the prey can't see it, and then the predator is able to get close and get the fish. Does that make sense? Everybody follow? Okay. The next major ocean jellyfish are one of the bigger classes of organisms that produce bioluminescence in the ocean. So what you see here are both pictures of jellyfish, kind of light images. If you went out with a camera in the ocean, this is kind of the picture you would see. If you turned off the lights, that'd be what you saw in the dark. Just in the paraphernalia, you get this kind of very starry image, and then in the uramphia, it just looks like a web. These things are thought to detract predators. These things are meant to look scary at night to keep things away. Have you guys heard of the anglerfish? Yeah. So here is the anglerfish. Now this lives down in the deep trenches of the ocean, and it has this little bioluminescent lure. Again, this is a bacteria that colonizes the light organ of this little lure, and it sits down there and waves it, and little fish will come and want to see what's going on, and it uses that to attack or to bring prey in closer, and then it attacks. Here, these are pictures from space, actually. So that's Africa, right around the Horn of Africa. You see that bright spot? That's just a huge pool of krill and dinoflagellates. So you can actually see the light from space produced in the ocean. And then right down Monterey, Santa Barbara, I believe that was taken about 5 years ago. There was a huge bloom of bioluminescent organisms off the coast of California. Pretty amazing that little organisms produce that much light to be seen from space. So bioluminescent organisms grow blue, which is high energy light. Does anybody have a guess as to why that would be? So you have the sun, and it's shining. Does light penetrate well through water? Kind of, sort of. So higher energy light is going to penetrate deeper. So blue light is going to be seen at deeper ocean depths. Most of the bioluminescent organisms are found at deeper levels of the ocean. So the light they would produce is mimicking the light that fish or other organisms down at that level normally see. So that's going to be blue light. Make sense? So we're going to move away from the ocean and talk about land. Again, classes of land organisms that produce bioluminescence. The firefly, click beetle, the railroad worm, mushrooms, bacteria. So are most of you guys from California? You probably all haven't seen fireflies really in your lifetime then. So growing up in Wisconsin, you go out by the lake or anything like that, like it seriously looks like a flashing light display at certain times during the summers when these organisms are mating. It's really bright and really flashes. And when I was a little kid, it was always fun to go get fireflies. You squish them, and the chemicals mix together, and you can make like green artwork with the fireflies all over the place. Kind of fun to do when you're like a 5-year-old kid. But so I grew up seeing that. It was really interesting. And then bacteria. are used in the worms, and the mushrooms are usually a bacteria creating the bioluminescence, whereas the firefly and the click beetle actually have evolved systems to make these, and I'll get into those here.
When you squish these, how long would the bioluminescence last?
The bioluminescence would last about 5 minutes, actually. It takes about that long for all the chemical that gets broken down by the enzyme to get eaten up over time. So again, bioluminescence is a form of chemiluminescence. I'll go through first here kind of how glow sticks work. Anybody know how glow sticks work? No? So you know when you get a glow stick, you pop it, there's that little glass ball inside of the glow stick. What's inside of there is a compound called diphenyl oxalate, that kind of two-ring structure there. And inside of the rest of the liquid in the glow stick is hydrogen peroxide. What happens is when that capsule breaks, the hydrogen peroxide breaks that chemical down into two phenols and that kind of box-looking peroxy acid. And this peroxy acid is a very unstable compound. What it does is it just breaks down right away. And during this breakdown, it gives off a lot of energy. Inside of these glow sticks, besides these two compounds, is a dye. And that energy is transferred to the dye. It excites the dye, and then as the electrons in that dye relax, it gives off the light. And the different colors are all caused by different dyes. So what is it in bioluminescence that makes this work? It's not quite as simple as the glow stick. So this was found to be an enzymatic process, and this is how it was done. What they did was if you took a clam, a bioluminescent clam, broke it up and put it in water, the water would glow over time and it would go out. Okay? Just for some reason or other, the light would go out. They then took a pulverized clam and put it in hot water, and the hot water, it went dark. Why do you think if you put pulverized up clam in hot water, it would be dark right away? Right. If there were proteins involved, the proteins would denature, fall apart, and no longer work. So that proteins are enzymes. So you take this mixture where the protein has been denatured and gotten rid of, and you take the other one where, anybody have a guess as to why that would go dark if this was an enzyme process? What's that? Exactly. You'll run out of the compound that's being broken down to turn into light. So you should have enzyme in that one, and you should have chemical in the other one. So if you mix them together and it's an enzymatic process, you should get light. And that's exactly what happened. So this was kind of the first experiment that demonstrated that the light production from a biological system was an enzymatic process. And what is this enzymatic process? They call it luciferase and luciferin. Luciferase being the enzyme and luciferin being the chemical. Do those names mean anything? Anybody want to? Yeah. Lucifer, the devil, light. Yeah, they thought they took this and that's what they named it, kind of giving it these names they thought was kind of a scary thing.
It's the same compound in fireflies.
Right, luciferase is the enzyme in fireflies, and that'd be firefly luciferase. Basically, any light enzyme is going to be called a luciferase, and they'll give it some surname before that. So what is the chemistry in this process? So you have an enzyme, luciferase, and a substrate, luciferin. The enzyme helps break down the substrate. In the case of firefly luciferase, you need energy source, oxygen, and cofactors such as magnesium salts. And this produces light. Different emission so there are all kinds of different colors of bioluminescence, from red to green to blue, and the different colors of light come from different chemical reactions evolved by those specific organisms. The first one we'll talk about is the firefly and click beetle luciferase. So the firefly's scientific name is Photinius piralis. And the reaction is like we just talked about, and it occurs in two steps. You have this compound luciferin, and in comes once it hits into the enzyme also is able to bring in ATP and magnesium. What's created is right away is a luciferyl adenylate. So ATP gives off two high energy phosphates, and the adenine combines with the luciferin. And this creates a lot more high energy molecule that's ready to be broken down. Oxygen comes along and breaks it apart and gives off CO2, gives off the AMP and some hydrogens, and creates oxyluciferin, and in that breakdown is the light output. Now, what's great about this reaction is it's amazingly efficient. So almost all the energy from the chemical breakdown in that bond is given off as light. Whereas compare that to an incandescent light bulb, of the energy pumped into that incandescent light bulb, 90% of that energy is turned into heat, where only 10% is turned into light. And this is about 98% is turned into light and 2% into other forms of energy. So it's very efficient. The other major enzymatic reaction that's used in bioluminescent imaging is a chemical called solentrazine. So this is from an organism called Rhinella reniforms, and it's a jellyfish. And this is Rhinella luciferase. And the compound here is called solentrazine. And this compound is very unstable, and just in the presence of oxygen, this enzyme, it breaks down to a solentramide and gives off light. And this light is blue or 475 nanometers, where I forgot to say the light from the click beetle and the fireflies is green to getting closer to red, the light given off. And both of these are used in biological systems. My favorite type of bioluminescence is produced by bacteria. And bacteria have their own bacterial luciferase, and they produce an aldehyde, which is basically a long carbon chain with a double bonded oxygen on the last carbon. And what it does, this enzyme takes aldehyde and a flavonoid, FMNH2, which, everybody ever heard of FMNH2? It's a reducing agent, like NADH. You guys will learn about this more later. But when these compounds are brought together with the luciferase and oxygen, the aldehyde is broken down through a series of intermediary steps to a fatty acid, and FMN is taking the reducing power off, and off you get light. And this light is also blue. What's nice about bacteria is they're able to produce, it's 5 genes in a bacteria that produce this entire process. The genes that make the aldehyde and make the enzyme, there's only 5 genes. Where the firefly, 1 gene makes luciferase, but the process of kind of making luciferin is not really well understood. So bacteria are able to give a bacteria the genes. You don't have to add anything. The bacteria just light up. With the firefly and the Rhinella luciferases, you always either have to add luciferin or add solentrazine to those when you're taking them outside of either the jellyfish or the firefly to cells to get them to glow. You just don't give them the genes and they glow naturally. There's a whole host of other luciferases and luciferin molecules. And you can see the structures and everything are different. There's the kind of ring one is dinoflagellates. Up top, that's solentrazine, the firefly luciferase. Down on the left, that huge compound is broken down by a luminescent mushroom. So you can see there's been evolved a lot of different chemical structures to make this process of light, which is very interesting that it's not one common ancestor. So what can we do with bioluminescence? Maybe we can have fun with it. So bioluminescent. So people have been making bioluminescent artwork. What that is a bunch of petri dishes with bioluminescent bacteria. And this stuff has been in art galleries and museums. Like, it's kind of been a big thing over the last few years, just kind of utilizing nature to make art. I think it'd be cool someday to have bioluminescent treats. You can make bioluminescent cakes or bioluminescent drinks. Maybe someday you could kayak in a bioluminescent bay. You go out and when you stir it up, the organisms that will be there will get excited, and you could kayak in a bioluminescent bay. Maybe more practically, we could use bioluminescence someday to make glowing plants to light roadways instead of using energy. They could have glowing pets if people really wanted. Anybody have any other ideas? No?
bioluminescent rays already exist.
They do, yeah.
I mean, there's a people, well, they call them phosphorescent rays. Down in Puerto Rico, for example.
Yeah, or you turn it up and the algae, yep. Mm-hmm.
There's no such thing as luminescence, right? Only luciferase, right?
Luciferase, mm-hmm.
And later, you said that they use luciferase to test if there was a living organism somewhere, and they would react to ATP. That's not the luciferase, right?
They just looked.
Well, they said something like they would test if there was an organism present, and they supposed to do something like spraying and then ATP something.
Oh, sure. So not only explain, they'd have to put in luciferase and luciferin, the compounds, but because that process requires ATP, living organisms are really the only thing that produce ATP. So yeah, by spraying these in, you can test to actually see if there was something living in that circumstance. Yeah, and that would be firefly luciferase, probably more than likely, because that is the brightest and most well studied luciferase. So how many of you have ever held a flashlight to your hand and see light through it? Yeah, everybody's seen that. So great thing about light is, even though you don't think about this, that light actually is able to pass through tissues. And which brings us to the next thing. Actually, we'll transition kind of from light production to what is bioluminescent imaging. So bioluminescent imaging, with bioluminescence, here's what we do. We either take a virus, a bacterial cell, a mammalian cell, or in some cases we just actually take the genes that make luciferase, and we either make the bacterial cell or mammalian cells glow by adding the gene, or a virus that's going to infect and has the gene for luciferase. We can take all these, inject it into a mouse. We have these special camera systems, we call them IBIS cameras, that have a very sensitive, the camera on top is cooled to, I believe it's minus 120 degrees Celsius. This cooling makes it much more sensitive, and it actually is able to count individual photons of light shooting up from the animal or whatever source of light you put in the box, and you can see where that light is coming from. And then we come down onto the computer, the data is compiled into a computer, and we are able to analyze the data. The amount of light usually correlates to the amount of cells that are there, and we are able to use this to answer a lot of questions. Bioluminescent imaging on the whole is a subset of a group of study called molecular imaging. And molecular imaging is the characterization and measurement of biological processes in living animals, model systems, and humans at the cellular and molecular level by using remote imaging methods. So, anybody have any ideas of other molecular imaging modalities? Has anybody ever had a CT scan or a CAT scan? An MRI? No? That's good, you're all healthy then. But these are all forms of molecular imaging. And here's a list of them. Ultrasound, animal MRI, bioluminescence. CT is like a CT scan is actually a 3D X-ray. You ever seen like kind of the images or models of like kind of skeletons that you kind of rotate and they on a computer and they look around, they look really nice. Those are usually taken on a CT scanner because it takes real nice 3D images. Micropet and microspect, you're actually injecting people with a radioactive compound that seeks out special cells, and the types of molecular imaging devices in that case actually look at the radiation being given off by these compounds to see where it is in the body. Those are used in cancer detection a lot of times. How does bioluminescence stack up to these other imaging modalities? So CT uses X-rays. We all know X-rays pass through tissue. Sensitivity detection, unknown. Like it's very, very, very good. CT scans. Ultrasound uses very high frequency, low energy sound, and you get that kind of grainy image. The sensitivity, you all seen out in ultrasound like a baby, how the image isn't really that good. It's because the sensitivity isn't real great. You don't get a good signal back. MRI, you're using radio waves, not high frequency sound. And an MRI is a lot better than an ultrasound because the higher energy usually means better sensitivity. The next one is nuclear medicine, like I was talking about, the radioactive tracers people put in. Now, if you imagine that's really sensitive, and does anybody have a guess as to why? Is there radiation around us? No, so the only radiation is coming from that little bit that's been injected into you, and radiation travels through tissue really well. So the only signal you're going to see in these machines is signal coming from the body. There's no background. And it's the same with bioluminescence. The visible light that is able to come through your tissue, light isn't coming from anywhere else if you put somebody in a dark room, so the light you're seeing is from the source that you're looking for, and it's very sensitive for that reason. So do we do this? We don't do this with humans or other people. Right now we work on small animals. So why do we work with small animals? One, with bioluminescent, light doesn't pass through tissue very well, but a mouse is small, so the light produced can travel through that small amount of tissue in a mouse. So dynamic in vivo assays inside of an animal are possible. Mice are a model system for human disease, so if we're able to study this, we're able to study a lot of the same diseases in mice, and the biology is very similar, so we're able to answer questions to human medicine in a mouse. We're able to improve statistics inside of a mouse. With people, if you give a bunch of people drugs, you have to give some people placebo and all that, and there's error from person to person to person. People don't necessarily take the drugs at the same time. With mice, we're able to regiment that exactly. You can give them a drug at the exact same time of every day, and you're able to follow them, control what they eat, and do all that. So you have a much better controlled system. And with the bioluminescence and everything, what we're able to do is whole animal imaging, so we get a lot more data from one animal. With these living bioluminescent imaging systems, you don't have to kill a mouse at time points to look inside of them and see what's going on. You let it live, you take a picture, you come back a month later, it's the same animal. So you're not going to get mouse to mouse variability because you're able to follow the exact same animal throughout the course of an experiment. So coming back to our basic strategy here, just to reorient you, what we need to do is how are we going to get this enzyme luciferase into animals, and then how do we get the substrate luciferin to these cells that have luciferase once they're inside an animal. So the first thing we have to do is introduce the enzyme. What we do is we take a cell, we give it the genes for luciferase, we add luciferin, and the oxygen ATP are just going to be present in living cells, so you get cells that produce light. So we all know here, just to orient you, the central dogma, DNA makes RNA, makes protein, right? Everybody's familiar with that? Because that's what we utilize. In a better schematic here, what we do is you have a cell. This is just a normal cell of the body. You have a nucleus producing with the DNA that produces RNA, which makes protein. And then you either have proteins that go to the cell surface, you have proteins that go to or stay inside of the cell cytoplasm or mitochondria. What we do is somehow introduce this gene, luciferase, in, and it becomes part of the DNA. So then, say we wanted to look at a gene that we think is important for cancer. So when it lights up, only cancer cells will light up. We'll take a gene that's important for cancer and fuse it with luciferase. So then this cancer gene now has luciferase when it's produced, and wherever it goes, usually intracellularly, like only cells that produce this cancer gene will light up. So we are able to utilize specific biological processes. Say you wanted to look at the liver. You can find genes that are only turned on in the liver. You can tag them with luciferase, and then you'd only liver cells would glow in the dark. We utilize this in all of our cases. Oh, I'm sorry. You have to always returning. When we talk about firefly luciferase and human cells or mouse cells, we always have to inject luciferin into the animal to get light, because again, we only give them the gene for luciferase. We don't give them all the genes necessary to make the substrate luciferin. And luciferin, so in my lab we go through probably a kilogram of luciferin a year. A gram of luciferin costs, I don't know, I think last time we were able to buy a gram of luciferin for about $400 a gram. So if we go through a kilogram of luciferin, we spend alone $400,000 just on luciferin to inject into mice to get light a year. So just to give you a perspective on kind of what this process will cost, and the cameras and everything are pretty expensive, and I'll talk about that too. So, just a little bit more into kind of how a gene is expressed. So genomic, like mammalian eukaryotic DNA has introns and exons. Do you guys ever heard of those? So only exons are what are expressed inside the cell. So what has to happen is your DNA is transcribed into RNA. The RNA needs to be processed, meaning the introns need to be cut out of the RNA. And then you have your coding sequence, which is translated into a protein, and then many circumstances, something called post-translational modifications, like some amino acids will link together even if they're not in the chain next to each other. Some of them will get chemically modified, and all that post-translational modifications make a functional protein. So in the case of luciferase, we've taken the gene with all the exons and introns from fireflies, and it's been really well studied, and they've taken out some of the introns. They've actually changed some of the DNA sequence in the exons to be more compatible with human cells and things like that. So a lot of genetic engineering has gone into this for us to make a brighter, better luciferase enzyme for in the case of human cells. So once we have this better gene, we need to introduce it into our cells. To do this, you have your promoter and luciferase. Anybody? I'm sorry, I forgot the last slide. What's a promoter? Did anybody tell me? No? Okay. So this is important. A promoter is the region in front of a gene that tells the gene when to turn on or when to turn off. So you can imagine your body, say in your stomach, you have genes telling your stomach cells when to produce more acid. Like after a meal, the acid gets washed out with the food. You need to upregulate acid production in the stomach. You'll have genes that'll turn that on. And what will happen is they'll have sensors on the cell surface that say, okay, we're low on acid, sends a signal down to the DNA that says, okay, turn on the acid-producing genes. And in this case, like the region that the chemical signal is sent from the cell surface down to the DNA and binds to what's called a promoter in front of the gene, and that promoter is what tells genes when to turn on and when to turn off. In the case of luciferase, a lot of times what we like to do is use promoters that are always turned on, like genes that are necessary for just a cell to live and produce energy, like an energy-producing gene, or a gene that that allows like cell membranes to be made and things like that. We'll put luciferase behind that because then the light is always turned on. Does that make sense? Okay. So we have our promoter luciferase gene, and we put it into cells either using a virus. So retroviruses like AIDS, when they infect a cell, they take the RNA, make it into DNA, and actually integrate into the chromosome. So they actually put DNA into your own cell, into your cellular DNA. So viruses are used. You can use chemical systems to put like a plasmid of DNA. Does everybody know what a plasmid is? Anybody heard that term? It's a circular piece of DNA that replicates itself and has the gene. So sometimes you don't want the enzyme or the gene luciferase to put into the DNA, but you want it in the cell. They'll do it on a plasmid. Electrical is another way to do it. You can actually shock cells with something called an electroporator with 2 to 15,000 volts of electricity in like a quick snap of the fingers. And for some reason, it's not really well understood. This makes DNA go inside of cells. So that's electroporation. So once you get the gene inside of cells, you have a Luc or luciferase positive cell. You add luciferin, and you get light up cells. So going back, so these are the kind of luciferase systems that are important for bioluminescent imaging. We talked about firefly. So that's luciferin is a substrate. It produces green to red light. Luciferin goes in the body and circulates for a relatively long time. You probably have signal for about 2 hours after you inject luciferin into a mouse. Luciferin is able to cross cell membranes, which is great. It can cross the blood-brain barrier, so luciferin is able to get to the brain, so you can look at brain processes. And it's able to cross the placenta. So if you're looking at developmental genes or developmental diseases of a fetus, you can use luciferase, firefly luciferase, and luciferin to look at. And the other thing that's amazing about both the firefly and the Rhinella luciferase is that these things are non-toxic. They don't even cause an immune response. These molecules are so small, and for some reason they don't, which is kind of a big deal, because almost anything you put into a person, inject into a mouse or a person, is going to usually cause an immune response of some type, but these don't, which is we take advantage of. Rhinella produces blue light. It's a lot brighter, but it only lasts a very short time. It crosses cell membranes, so it gets inside of cells, but we don't know if it's able to cross the blood-brain barrier or placenta because the studies are so hard to do because the enzyme and compound kind of use up so fast that it's very hard to do biodistribution studies, so we don't really know a lot about it. But if you want to look at something right away and in a bright system, we use that Rhinella luciferase. And bacteria are great because they produce their own substrates. So you don't have to worry about injecting these substrates into a mouse and seeing if it gets where it needs to go. The bacteria just light up on their own, are always glowing. So how sensitive do you think this is? How many luciferase cells do you think you need to be able to see the light? Any guesses? Take a guess. 1,000? Good guess. Well, believe it or not, using what we call the SV40 promoter, which is a viral promoter that when this virus is inside of cells, it always is growing. So we use it. It makes things bright. Using our special camera system, we can see 2 cells. 2 cells produce enough light to be detected by our camera. And here you see a graph of how many photons of light per second are given off from basically 2 cells, 10 cells, 100 cells, 1,000 cells. So at 2 cells, you're giving off about 10 photons of light per second per cell, which is pretty amazing. That much light is coming from these things. And it's also pretty amazing that we have cameras that are able to detect that low level of light. For you to be able to see something, you're going to have to be at about 10 to the 7th, 10 to the 8th for it to be visual to your eyes. So these cameras are a million times more effective at seeing light than our eyes are. So we've talked about kind of making bioluminescent cells and everything. So the next step is what animals, what stuff do we use? Oh yeah.
Does it change the behavior of the cell at all?
So because that ATP is being used, there is a little less energy available. But in our studies, only in some certain bacterial circumstances you'd have to infect with a few, like maybe 20% more bacteria to get an infection. But in cells and tumors that we make glow, it really hasn't seemed to have much of an effect, which is, again, a pretty amazing feat. So, what type of mouse do you use? Anyone have a guess of which mouse up here would be the best? Hairless. Why? Exactly. No hair for the light to have to go through. So that's a special mouse. We call them nude mice. Nude mice have no thymus, and therefore they have basically no immune system, so those aren't the best to study, but for just light purposes, that is the best one to use. Next, which one do you think would be best? White, because white hair does not absorb. White doesn't absorb light. So black would be the worst, gray, brown, white, and then the nude in that order. So we try our best to use white or nude mice for our studies. In some circumstances, we have to use black mice. And in those circumstances, what we actually do is, you guys know what nair is? We take nair and nair the mice to make them, to shave them. So we get better light penetration from the cells. So here's the basic principle in using mice is you have a bioluminescent source inside of the mouse. The light is scattered and kind of comes out and funnels up towards the imaging system, and you have a detector underneath behind the lens that focuses the light. So the amount of light the camera is detecting is dependent on a lot of different things. The source brightness and shape inside of the mouse, how deep that source is inside of the tissue, what wavelength that light is produced, what wavelength the light is produced will matter how much light comes through. The quantum efficiency of the camera, how good it is at detecting the light, how good the lens in front of that camera is, the skin and fur color, the curve of the mouse, actually the shape of its body will have an effect on how much light comes through. And then background emission of live animals. So believe it or not, alfalfa is used in mouse chow. And alfalfa, if you put an alfalfa piece of mouse chew out in the light, it's fluorescent or phosphorescent, and will take in a bunch of light, and over time the alfalfa chow will kind of give off light. And the camera is sensitive enough to detect this, so it's best in a lot of circumstances to actually feed mice alfalfa-free mouse chow so we don't get any background light even from food, or the fecal pellets after the digestion. Those will glow still from that light that was gathered in the mouse chow to begin with. So what color light, do you think is best for imaging? Red, exactly. And here's a great example, and hopefully you guys can see this. So here we just use a white flashlight. You can see it reflects off the skin. Well, this is just a finger, and you can see it goes through. It's hard to see here, but you have green laser. It doesn't really reflect. It's all kind of absorbed.
You can turn the light if you want.
Oh, great. There we go. So the green, basically there's no reflection even. All the light is absorbed, but none of it is able to transmit through. Same with the blue, but the red reflects a lot, and you get a signal almost as good as the white light transmission. So in bioluminescence, it's best to try to use red light sources, enzyme substrate pairs that'll produce as far to the red as possible. So firefly and click beetle produce green to orange light, so that's why we've kind of modified the gene through genetic engineering to kind of make more orange light, so that's what's used in most circumstances. That Rhinella luciferase is a blue light, but it's very bright, so that's how that, even though it's good at penetrating through tissue, it produces enough light that we can see it. And the bacterial luciferase is blue, but again, in bacteria, you're able to usually, if you're infected, there's going to be billions of bacterial cells inside of you, so that's enough that they produce enough light to be seen. We'll go through what absorbs, why that color light is best. So the major absorbers inside of cells are proteins and melanin. Melanin is the skin pigment. And then there's scatterers, such as mitochondria and nuclei. Light hits those and it'll get scattered because they're small molecules. But the major absorber is hemoglobin and heme in our blood cells. So hemoglobin, here's a chart of absorption and emission. So the gray bar you're seeing here, this is where light is absorbed by hemoglobin. As you can see, the blue light, the Rhinella luciferase, a lot of the light produced is in this absorption range, whereas the green-orange firefly luciferase is kind of outside of this hemoglobin absorption. So again, that's why we like to use firefly luciferase. The light produced is of a higher wavelength than hemoglobin can absorb, since hemoglobin is the major absorber of light. And we like to call this area out here, away from hemoglobin, the tissue transmission window, the areas where light is best transmitted through tissue. That make sense?
You guys have also heard of that as the optical window.
Okay, I'm going to talk a little bit here about the cameras, and then we'll take a little break. So what do we acquire the imaging with? So we use a low light detection system, and these are called IVIS cameras, or in vivo imaging system cameras. They're produced by a company, or were produced by a company called Xenogen Corp, which is just bought by a company called Caliper Biosciences. And these are kind of what the newest models look like. When I first started, this is what we would get. You get a cryogenic refrigeration unit, which cools the camera. You get a camera controller, which would focus and run kind of the hardware and software for the camera. You'd have an imaging chamber, and then you have the computer, which you can control everything from. Basically, all this imaging chamber is, it's a spot where you can pump in anesthetic to knock the mice out, so they lay still, so you can image them. And it's just a dark box that's heated, because mice, when we study mice, we want to keep them as comfortable as possible. We want to make sure we're not causing harm, so we use a heated stage that keeps that body temperature, so when they're under anesthetic, they're comfortable. So this is just a dark box with a heated stage that has a camera sitting on top of it. Again, like I said, the camera is very sensitive, but it has to be cooled to minus 120 degrees Celsius. So we use the cryogenic refrigeration unit, and the computer controls everything. And then now everything comes in one tight, little tiny package. And this thing will run about the new best models are going to be around $1 million. So for one camera. So what kind of pictures do these cameras produce? So the first picture, it always just takes a light image, just a picture of the mouse laying in the chamber. Then what it does is turns off the light that it used, the flash that it used to take this picture, and just, for as long as you tell it to, collects the light. And this is a low light image collected in the dark. So every speck that you see is light coming from the mouse above just kind of cosmic radiation and any background that would be there. Red intensity, meaning the brightest light comes from there, where blue is the lowest intensity. And then what we do is you take this pseudo-color low light image and overlap it with a reference image, and here you have exactly where the light is coming from in the mouse. You have the superimposed image, so you know, okay, the light in this mouse is coming from around the kidneys, kind of around the back. It can guide you. If you want to know the exact organ, it kind of gives you a general idea. You can assume, or what you'll do is you actually can open the mouse up and expose the different organs, and you can see. It'll lead your studies. And in mice, how sensitive is this? So comparison, it takes a few more cells to see it in a mouse. So say we just injected the cell, luminescent cells, into the stomach cavity of a mouse. To be able to see it, you really start to need to be above background. 100 you can barely see, but 1,000 cells you can see pretty good. And again, you can see as every log increase, you get about a log more light at that point. It's a linear thing, but you need a lot more cells to be able to see it in a mouse, because that light has to travel through the tissue. You can see here, what we've done is taken cells and had the different cells of the body express the luciferase. And in vitro, so just cells in culture, if the skin is producing the signal, if the lung is producing the signal, if the liver is producing the signal. So liver is the worst, because liver has a lot of red hemoglobin, it's dark colored, you get the less light. Lung, a little bit better, but it still has to go through like a muscle layer. Skin, you're above the muscle layer, there's a lot of blood around to collect light, so you get the brightest. And then again, in culture, cells in culture are the brightest. Here you can kind of see the images we get. So this is just infected skin on its back. You can see the lungs, if you're laying the mouse on its back, how the light comes through, and then the liver. The light localizes pretty good to the target organs. So next topic we'll do is we'll talk about how we can image with bioluminescence. And to leave you here, I'm going to leave you just with before break, a little video of kind of what we can do. And what we did here was infected the mice with a virus that infects liver cells, and we kind of follow this liver infection with luciferase over a course of 6 days. So what we do is the mouse is actually running around in a cage and everything. What we do is when we want to image it, we just take it out of the cage, put it in a chamber with some anesthetic. It falls asleep. We lay it down on its back in the chamber, take a picture of it for about 2 minutes. It wakes up about a minute later after we take it off the anesthetic, let it run around, come back and do the same thing like an hour later. So the mice, this system it used to be a little bit more difficult, but we use just a gas anesthetic, so we just put a little nose cone on there, on them, and they just lay down and suck the gas down, and they're fine, and they wake up a minute later after you take them out, and you seem a little woozy and dazed, but they're fine. So, any other questions? Yeah.
I don't know if it's very much well, but like one of the end slides where it had it with how many photons it was showing, was like when it had very few photons, it was like showing more in like its nose area, but then when it was like higher in like its stomach, it didn't have that. Is that from?
So the nose, what you guys actually saw, I don't know if you noticed in those pictures, and let me see if I'll go here. We'll go back. But what we try to do, I don't know if you can see, but there's a little bit of a nose cone there, like a little plastic kind of ring around the nose in this mouse. So when we first started doing bioluminescent imaging, these nose cones actually were a problem, that they would sometimes give light off, and we worked with a lot of different plastics and things like that to find compounds that didn't do that. So that nose light was actually, the light that's coming from that is, the background in those is so low that there's a little bit of light coming from the plastic, and that's what you're seeing from the nose area. Anybody else? Okay. So yeah, we'll talk about what my lab, at least, some of the research projects we've been doing, and what we use bioluminescent imaging for. So we all know now the basics, how we are able to use bioluminescent imaging here, right? Okay. So the first thing I'm going to talk about is how we look at investigating cancer with bioluminescent imaging. So we have in our lab what we call cancer cell lines. Cancer cell lines are taken from cancer, and we're able to grow them in culture dishes, and just like kind of you're able to grow cancer cells or a cancer in a dish. This is great because then you can take this cancer and implant it into a mouse. You don't have to wait for mice to just spontaneously get cancer and then study it in that way. You're able to give them cancer. I know, not the best, but what we do is we take these cancer cells we grow in the dish, and we use the promoters there are constitutive, so cells that are always going to express luciferase. We introduce the gene there, and in this case, what we did actually was gave the mice breast cancer. So in mice, they're like cows or whatever. The mammary organs are down kind of by their bellies. So we gave them primary breast cancer. What we did then was after they had a tumor down there, we took that tumor out. We went in and did surgery and removed the tumor we gave them. Have you guys ever heard of metastasis, when cancer spreads or metastasizes? So because we gave them bioluminescent cancer cells and we removed that tumor, you don't get light there, but over time, this is about 2 months after we took the tumors out, all of a sudden you start getting bioluminescent signals like up in the lungs and things like that. And this is from like one or a couple cells that broke off from that original tumor, somehow got into the blood vessels and were able to go around. And the most common place usually for metastases is the lungs. Anybody have a guess as to why? Yeah.
The blood gets circulated back through the lungs, pick up fresh oxygen, so the lodge there.
Yeah, exactly. Very thin, like small capillaries there. And if you have a couple cells, like a clump of cells, they're going to get stuck in that spot, and they just kind of find a happy home where they're going to have oxygen and blood flowing around, plenty of energy, and sugar being given to them. So kind of a happy home for them, and they're able to grow in the lungs. So lungs are a big spot where you get metastases for that reason. And here we're just showing that we can actually see these, and these metastases in these cases are very tiny, but they're bright enough that we can see them kind of before you'd actually if you even cut up the lung, to actually be able to see these metastases. So it's a good way to study metastasis at an early time point. Here you just see the kind of the gross anatomy of kind of where you'd see it before, and I know it's really hard to see, but that's one like kind of healthy lung, and then a lung with kind of nodules on it, and that's like the lung, what metastases would normally have looked like, how you would have tested for them in the past. So this bioluminescence was a great way to assay this type of biology earlier in time. Yeah.
The second mouse is dead.
You mean, is this mouse dead?
Deceased, yes.
So in just as far as like mouse and kind of bioethics go, we at Stanford, kind of part of what we do is if mice become like really sick and like they could survive, we will sacrifice them, we'll put them down. We don't let them like get diseases that are so crazy and bad that they're like suffering awfully. So actually that mouse, if you just saw it running around a cage, would look healthy. Like those metastases are pretty tiny. The reason it's all this blue light is I think that mouse, if I remember correctly, the metastases were really close to the rib cage. They were like kind of in the front of the lungs, so they were up closer to the surface, so the light was just brighter from that spot. So not just breast cancer, but lymphomas, colon cancer, breast cancer, ovarian cancer, brain cancer, cervical cancer, liver cancer have all been studied using bioluminescent imaging. So another great thing we can do in studying cancer is look at how good cancer drugs are in a quicker, better fashion. So again, we implant cancer cells into mice, and here we're studying to look at two different drugs. So 5-FU or 5-fluorouracil and cisplatin. So 5-fluorouracil is a pyrimidine analog. You guys know what a pyrimidine is? So DNA is purines, and pyrimidines are the A, T, C, and Gs. 2 are pyrimidines, 2 are purines. So this drug looks a lot like the pyrimidine bases, and it kind of, when DNA is made, it kind of fits into that pocket. So when DNA is made, this compound goes in there. So DNA is made incorrectly, and those cells die. So this is a chemotherapy. Fast-growing cells, if you give this the fastest-growing cells, are going to take up the most of this. When they're replicating the DNA is going to get made wrong, and the cells die. So this is like actually one of the first big chemotherapy drugs out there, and it's been around for about 50 years now. That's 5-fluorouracil. And cisplatin is a platinum drug, and platinum usually is in our bodies. It's a very reactive kind of metal in cells, and once it's inside, again, like fast-growing cancer-type cells, these platinum complexes bind to DNA and make DNA kind of link upon itself, which, again, that causes cell death. So that compound is used as a chemotherapy. So you're probably thinking, well, these probably work against normal cells too, and they do, but that's why chemotherapy has a lot of bad side effects and things like that. But what we're looking at here now is how well these drugs worked against a cancer that we experimentally gave a mouse. So here we have a PBS, which is just, PBS is phosphate buffered saline. It's just a buffered solution that when you inject it into mice, it doesn't cause them to get sick or anything like that. You can't just inject pure water into a mouse. It'll actually get sick because it'll cause like osmotic shock where you just inject like distilled water in. So that's why you always use like a little buffered salt water. So you can see that's the number. It's 0, 7, 14, 21, 28 days. So 4-week study. We gave the cancer here at day 0 and then started giving them drugs. You can see within 2 weeks and then 4 weeks that basically the signal is maxed out. Like all you're seeing is red in the PBS control. So the cancer took over pretty good. The 5-fluorouracil, here you can see that one mouse, it kind of prolonged maybe the life or cancer progression in a couple of the mouse, but the cisplatin looks like it worked the best. That makes sense? There you see how this is an easy way. What you would have had to do in the past is for every one of those time points that you're seeing, you would have had to have killed probably 5 mice. So pull out their tumor and then look to see how many tumors there were in the lungs and how big they were and things like that. And that was the way you say it. Here, this is just 9 mice. And you're able to follow all 9 mice. You're just able to take a picture of them at every time point. So you save a lot of mice in doing this process, and a lot of labor and work, because like dissecting and like looking for tumors through like an organ is a very time-intensive process.
So, quick question.
Yeah.
So 3 conditions enough? I mean, because you're doing with cisplatin and without cisplatin.
What we normally do, what we've come to find is better is 5 mice. 5 mice is per condition is great. In most cases, there's some of the stem cell stuff, which I'll get into. We found maybe a couple more because weird different things seem to happen in those cases. But in cancer and infection, 5 mice per group usually is very statistically relevant. You don't get a lot of outliers with 5 mice. So the next therapy, so that last one we showed was looking at cancer with established therapies. Both those drugs have been around for a long time. So here, what we did, a member of the contact group took blood cells from like a mouse and put them in culture and tried to grow them. But in doing so, added different like chemical signals that our body produces to create kind of a super immune cell. We call these cells cytokine-induced killer cells because the chemicals that we add to the blood cells are called cytokines. And what these cells do, what's amazing about them is when you inject them into a mouse, they go straight to tumors. There's something about them in this process that matures them. These cells go and home to a tumor site. And that's actually what you're seeing here. Here's a mouse with no tumor. They injected the cells, these cytokine-induced killer cells, in the tail. So there's a vein that runs through the tail. And you know how like any of you guys had an IV, like they put it into your arm vein? So the mouse vein, we inject in there, and that's kind of like the equivalent of doing an IV injection in a mouse. We do it into the tail vein. So 10 million cells of these cells were injected at an hour, and you see that's just the nose cone type thing that we're seeing there. At 24 hours, you kind of start to get some signal here. You get some signal in the tail and in the feet. Why do you think the tail and the feet would have signal? What's different about the tail and feet on a mouse from the rest of its body?
No hair.
No hair. Exactly. So these cells are just kind of swimming around in the blood, and they have luciferase in them. But here, there's no hair. That even though white hair, it does not absorb. It scatters the light. So just kind of since it's going around the blood, the skin there, you're able to see it. But then at 48 hours, like you start to get a much brighter signal, and if we follow this along, basically all the signal goes right to that tumor site. So these cells just go and collect and go to attack the tumor. And we use this process to study these cells and how good it was at killing tumors. And it worked really well. And then what we did, or actually Steve in our lab did, he took vaccinia virus, which is a little bit like smallpox. You guys all know smallpox, like really bad virus. Took vaccinia virus and mutated one gene in it that is a gene that makes, when the virus infects a cell, makes the gene or makes the cells produce a lot more of the DNA bases. Because the DNA bases are, if you're going to make a lot of your virus, you need a lot of bases to make a lot of DNA or RNA around. So he mutated that gene, so the only cells that'll have a lot of bases around are cancer cells because they're really fast, rapidly growing. So he takes these CIK cells, gives them this virus, then infects a mouse with them. These cells go to the tumor. The virus kills some of these CIK cells, gets out. Now it's right inside of the tumor. They infect tumor cells, and the virus then kills tumor cells. So not only are these cells killing the tumor, but the virus and the cells work together to kill tumors. And this is a really big deal. Some cancers in mice that are known to like basically go from no tumor to death in like 10 days, really fast, serious cancers. This worked. It like cured mice at like day 5. So it's a big deal. I think this year they've just progressed it along. They're going to start like human trials of this therapy. So it's a really big, cool deal. Hopefully it works. And we use bioluminescent imaging to study this and move it along way quicker. Does everybody understand that? So in both of those cases, we just took cells in culture and transfected the gene into those cells, and we had glowing cells. What if we wanted a mouse to do this? We needed to create a transgenic mouse. And how we did that was we created a transgenic mouse, and what we did was took the promoter, the special promoter we use here is called beta-actin. So actin is kind of the skeleton of all your cells. So every cell in the body produces this beta-actin. So we use the promoter from that gene and put the luciferase. And as an embryo, they injected this into the mice, and the mouse came out. We had which were kind of spotty. Only some of the cells of the embryo took it in, so you had kind of like a cow-looking mouse of bioluminescence, right? It'd be spots all over it. And then we got lucky that of these spots, the genes that were responsible for making the sperm and egg from those males and females had that area of the development got this gene. So then the sperms and eggs that those mice were able to mate had the bioluminescent genes. So then we mated them, and we have now mice that every cell in the body produces luciferase. So we inject them with luciferin, and they just glow. They glow bright. Every cell, and we use these cells, and I'll get into that. One of the things that's important to think about is when you give mice luciferin, it has to get to the cells. It needs to, in order for those cells to create light. So here's an example. We have this universal donor, so every cell in the body produces light, and we inject IP, which is into the stomach cavity, intraperitoneal cavity, and then here we have luciferin injected IV into the tail vein. And you can see that when we injected IP, the signal kind of all stays in the stomach cavity, right? Because we injected it into the stomach cavity, it's hard for it to get out there. It will absorb in, and over time that signal will go places, but this is right away. Same amount of time after injecting IV, and you can see the luciferin's gone everywhere in the body, and you have a mouse, or you have you have signal from every cell basically in that mouse. And this is important when you're planning your studies, which method is best. This takes about 5 seconds a mouse to inject. This takes about 5 minutes, to get the mouse and find the tail vein and inject it in. Where this is just, you pick the mouse up, its stomach is right there, you just go in, inject, and it's really fast. So usually with this, if you wait 5 to 10 minutes, the signal will be everywhere, but sometimes you need that signal to be right away, and then you have to do IV. So that's an important consideration when you're adding the luciferin to the cells, or to the mice. So in this transgenic mouse where every cell in the body is bright, there is some differences. So this beta-actin promoter, even though it's expressed in all cells don't express that gene equally. So here we have populations, so helper T, killer T, B cells, NKs, natural killer cells, macrophages. What kind of cells are those? Anybody? Those terms kind of mean anything? Immune cells, right? Like blood cells. So here you can see, so this is the same amount of cells in every case, but macrophages are way brighter than helper T cells and killer T cells. So this is a consideration. Well, you always have to know, if you want to look at a cell type or something like that, you always have to do this analysis and see how much light is coming from those exact cells you're looking at, so you can understand and say, quantify how many cells are probably in there, because that's a question you're always going to get asked. So just showing, even though we've given the same gene and every cell has it, the light can be expressed differently. So there's always some considerations to think about when you make these mice or you do these experiments. So how did we use this mouse? We have a mouse that produces light in every cell. Well, these rare kind of cell populations are important in biology. So your bone marrow produces basically all your blood cells, and you get red blood cells, you get your white blood cells, you get your platelets. And inside of the bone marrow are things called stem cells. And what we first wanted to look at is, you guys have heard of people needing a bone marrow transplant or things like that. We wanted to look at that process. Like after you give a bone marrow transplant to somebody or a mouse, like how long does it take for the cells to kind of populate the body? So what we did was we took that Luke donor mouse, the bright mouse, we take out its femurs, break the bone open, and you basically just scrape all the cells out of the femur of the sacrificed mouse. You get the positive, the glowing bone marrow cells, and you inject it into a donor or a recipient mouse, excuse me. You add luciferin, and here we follow this mouse over time. The injection, in this case, was into the spine. You get this little signal here at 1 day. So this is just 1 day in, the cells kind of are hanging out where we injected. At 4 days, you can see these cells have started growing. They're kind of, like they're in the major bones, right? Here's the femurs in the spine. This is the spleen. Like when cells are produced, they grow, and like a lot of the immune cells go to the spleen. So that's what you're seeing. These are the lymph nodes in the neck. So a lot of immune cells there. You can see like at 4 days already, the population of bone marrow cells you get is already kind of gone everywhere. And then at 7 days, basically the whole mouse, the blood system, the hematopoietic system of the mouse is basically all bioluminescent cells. So it only took, at least in the case of mice, about a week for this bone marrow to take over. And how would you do this? Did anybody have an idea of how you would even think, how you'd look at injected cells if they weren't labeled in this way? It'd be very difficult. What you'd have to do is find a mouse that is very similar genetically but has maybe a different marker, and what you'd have to do at every time where you'd have to kill mice, drain their blood or take out their bone marrow, and then try to assay for that different marker on each of the cells that you've taken out of the body. And that would take tons of mice, hours upon hours of labor, and this took 5 hours and 5 mice. So again, this is very powerful and quick and a great way to study these processes. So once we did this, we just gave whole bone marrow and looked at how quick it was. Now the big thing, a lot of us know, like there's a lot of research and things like that going into stem cell research. And not only embryonic stem cells, but there's something called hematopoietic stem cells. And these stem cells are the stem cells in the blood, in the bone marrow, that are able to create every other cell type. So here's a little schematic. So you have your hematopoietic stem cell, or HSC, which either self-replicates or creates the next step down, which is a multipotent progenitor cell. And this starts breaking down, and you get outgrowth of T cells and B cells and macrophages and other white blood cells, red blood cells. But only that hematopoietic stem cell is able to give rise to all the other cell types. So we did a very similar experiment. We took the luciferase donor mouse, took out the bone marrow, and by cell surface markers, we were able to just isolate out I believe this was 2 hematopoietic stem cells. So we took 2 cells from the bone marrow that were able to grow and give rise, injected them into a non-bioluminescent recipient, added luciferin, and you can see here, in this case, these are different mice, but those cells, after they were injected, somehow found their way into the femur, started growing a population of cells. Day 12, day 17, basically that whole femur now is controlled by the new cells that you gave it, the bioluminescent cells, and then again the whole process kind of gets faster. The whole body is repopulated by these bioluminescent stem cells that you gave it, the blood system is. In this case, and this is something we don't really understand, the cells got there. They're not in the femur, but they're in the forearm bone, in the bone marrow of the forearm. But in this case, they just sit there and grow and kind of stay as hematopoietic stem cells. They don't start to really grow into and proliferate into cells that are going to repopulate the blood supply. So in this same experiment, we were able to kind of tease out 2 different things. Stem cells, hematopoietic stem cells can come in, find a spot to live in the bone marrow, and then grow and create progenitor cells and create cells that are going to produce all the different blood cells. or they just come and just kind of keep making more of themselves and don't create the cells that are going to repopulate the rest of the body. And why that is, it's something that we're trying to figure out now, like what the difference in those two situations was that allowed one cell to go crazy and one cell to kind of not do anything. Does that make sense?
And are all these mice genetically identical? Are they always cloned?
Yes, all these mice are inbred, have been inbred for 20, 30 years, and are supposedly genetically identical. Yeah. We use, in our lab, we always use female mice, not male mice, because female mice are a little nicer. Male mice try to bite you a little bit more, and they're a little harder to work with. So we just always use female mice, and that also is something when you're thinking about experiments and doing controls. If you're using all female mice, that's just another variable that you're taking out of the system. So we looked at cancer, we looked at hematopoietic stem cells, we looked at kind of drug discovery. We'll do a little bit more about drug discovery now and look at gene therapy. So cystic fibrosis. Anybody know cystic fibrosis? Heard of cystic fibrosis? So it's a genetic disease that causes a dysregulation of a chloride ion pump in the lungs. And basically, this mutation, you get thick mucus, people get Pseudomonas infections, and it's chronic, and the life expectancy is now, it used to be you'd only live to your teens, now people can live upper 20s, early 30s with like the therapies that exist, but really it's a genetic defect, and we're not curing the genetic defect, we're just always curing the symptoms that come about because of the genetic defect. Gene therapy is something that maybe we could introduce a good copy of the gene and see what happens. So we use bioluminescent imaging to kind of study this process. Do we have an idea of how you'd introduce a gene into, say, the lungs of a person? What you'd use? A virus, exactly. You'd put in, we all get colds, something that has probably come in like the flu or something, it infects the lungs, you get a disease. Let's take advantage of those types of viruses and infect you, but have them carry genes that are going to be beneficial. So it's exactly what we did. We create, we use an adenovirus, it's called in this case, that we give a copy of the luminescent gene, and we want to see what happens. So we inject this luciferase containing adenovirus into the animal. And you can see here what we did. So this is 4 different groups. Adenovirus plus a compound called perfluorocarbon, adenovirus with no perfluorocarbon, and then AAV2 is, let me see here. I'm sorry, this is supposed to be AAV2, not AAV5. AAV2 is the control virus with no bioluminescence. So we always want to control to make sure. And what you see here is only when we give adenovirus plus this perfluorocarbon. What a perfluorocarbon is, it's a gas, it's a heavy gas that when you give the virus, you also have the patient, or the mouse in this case, inhale this down, and it pushes the virus down deep into the lungs. So you get the virus takes better, infects better if it's able to get down deep into the lungs. And so we were looking at ways of delivering virus more efficiently, and you can see with perfluorocarbon, the virus does way better, whereas here, kind of 10 days post-treatment, the signal already starts to fade away if we don't use that perfluorocarbon. So this is a way we're able to look at possible ways of enhancing, making gene therapy better by evaluating the therapies associated with the gene delivery. And the problem also is using this virus, there's a problem with, again, the sustained delivery, and that was that over time the signal decreases, the cells don't stay infected. And here at time 10, you can see the difference between perfluorocarbon and non-perfluorocarbon. Again, I'm sorry I messed the labels up. But the AAV5 with perfluorocarbon is much brighter. Okay, now let's talk about using bioluminescence to study infectious disease. So bacteria, I talked about this. You don't need to add luciferin to a mouse to get the signal. The bacteria glow. They produce their substrate and the enzyme all alone. So it's a 5-gene operon. Lux A, B, C, D, and E are the 5 genes. A and B make the luciferase enzyme, and C, B, and E are the genes necessary to make the substrate. We put this in using this special plasmid, not important, but what can we study? So in the case of listeria, we made the bioluminescent listeria, and I talked about, we saw this signal. This is like 20 different like a bunch of different mice, at the same time point, you always get this signal coming to that gallbladder. So we wanted to verify that was the gallbladder. We took the gallbladder out, all the signals in the gallbladder. We squeezed the bile out of the gallbladder. It grew it on a plate. You can see all the bacteria that grew out of it are bioluminescent, meaning it was probably listeria that were in there. And then we did a microscope image, used the bioluminescence to guide us to standard techniques here now, where we're using just light microscopy and a gram stain to look at the bacteria, and we can see that these are in fact listeria growing, and they're growing this chain morphology in the gallbladder. So without bioluminescent imaging, again, you would have had to have just decided one day to take a mouse that you infected with listeria, take out its gallbladder for whatever reason, and then look at it and see if listeria were there. Really, there's nobody ever thought bacteria would grow in the gallbladder. So without bioluminescence, I don't know if this would ever have been found, or not for a while yet. So we use the bioluminescence to guide us down this path. Another model, kidney infections can exist from, if you get a bad bladder infection, it can sometimes go up from the bladder into the kidneys and cause pyelonephritis. Here what we're doing is using uropathogenic E. coli, bioluminescent, that we inject up the urethra into the bladder of mice, and over time they get kidney infections. We're able to then look at drugs to study this kidney infection. Another one, people who get bad wounds or burns get Pseudomonas infections in their wounds or on their burns. Here we're looking at the ability of a compound called enterofloxacin to be used as a therapy against the Pseudomonas wound model. So we wound the mice and infect the wound with Pseudomonas. You can see the control that are just given, again, the PBS, that buffered solution, because you always want to give them something as a control. Kind of stay bright and nothing really happens. You can see the quantification of the dark bars. And then when we give this compound enterofloxacin, the mice get cured. We lose the amount of bacteria. And without bioluminescence, the way we would have done this is every mouse would have had to have been killed. You would have kind of isolated out the wound, kind of macerated it in like a buffer, took some of that out, plated it, and counted the amount of colonies, bacterial colonies that would come. Here, you're able to do it very quickly. With here, I'm only demonstrating 2 mice, but we used 5 mice, and it just is a much better method to look at this. Okay. Here, what I want to talk about is future directions for bioluminescent imaging. What we're going to do to make this better. And we'll all go back. Remember, we had this window of if we got light here, we'd have a much better signal. It wouldn't absorb through the tissue. You'd get a lot better signal out of the mouse. So what are people doing? Are they actually making different luciferins or mutating the luciferase to try to shift the light more red? So it's going to transmit through tissue better. And here you see this is a solentrazine mutant, the Luciferase, where they've changed some of the amino acids in the active site of the enzyme, and you can see a much brighter signal. Here we have firefly luciferases where the active site has been mutated, and you can see this is now the emission spectra, which shifted from about 560 nanometers to about 613 nanometers, which every few nanometers we go is going to be better signal and better signal. So people are trying to genetically engineer the luciferase enzymes to make a better signal. The other thing people are trying to do, because of this luciferin addition problem and the cost associated with it, are trying to, trying to modify the bacterial 5-gene operon so that that can be introduced to mammalian cells. So then mammalian cells will make their own substrate, make their own enzyme, and express light. It's been problematic. They've gotten some yeast to do it, but it's never. We think that aldehyde, the compound that the substrate in bacterial luciferase, is actually toxic to eukaryotic cells in most cases. So that might not be the best mechanism. The next thing, is in fireflies, we believe there's an enzyme. Nobody's proven this yet, but we believe there's an enzyme that takes oxyluciferin and turns it back into luciferin. So it does the opposite of luciferase. And if we can introduce that gene in with luciferase, we'd be able to recycle the luciferin. The signal would be sustained. You wouldn't have to keep introducing luciferin. But again, this has kind of been a wild goose chase, and nobody has found this gene, or has at least been able to make it work if we have. And finally, the next big thing that we want that is being done is there are limitations to bioluminescent imaging, specifically getting light through tissue. In humans and us, it would be very difficult to put us in a camera, lay us still, and hope that there's enough cells in a spot to get enough light to transmit, because we're much thicker than a mouse, so the light transmission would be tough. So maybe we can use bioluminescence in conjunction with these other methods like MRI or the nuclear medicine, radioisotope stuff, and in combination, we'll be able to answer your questions. Maybe use lower radiation levels from those methods and enhance it with bioluminescence. And then we're almost out of time, so I just want to make sure I get you guys. I guess here, we'll give the take-home message. So what I want you guys to impress upon you about bioluminescent imaging, all you have to take is that the standard method was you could do a lot of things in culture very easily. You can look at cells, you can assay for their cell surface markers, you can learn a lot about them, but there's always been, if you put it into a mouse, it's always been this black box, and to get something out of that black box, it's been very difficult, very labor intensive, and takes a lot of mice and a lot of people hours to make it work. You use a lot of different techniques on those tissues and things to extract that are expensive, like microarray or pathology, or crude measures like looking at the size of a tumor and saying that's how much tumor there is there. And the data, because of that, is weak or isn't as good as it could be. With bioluminescent imaging, we view it as a single reporter, luciferase. We can put it into this black box, and now we have light. It guides us to areas where the cells are, where the biology that we've kind of put into the system is ongoing. We can then take that system out and take it and look at it in much more detail, because we've been targeted to the areas where the system is working correctly. And in that you have intact animals. You're using less animals. It's real time. And you can predict, using bioluminescence, like the ability of like a drug to work. So with that, I just want you guys to take that home. I want to thank everybody in my lab, Dr. Jen Prescher, Steve Thorne, Yuan, and Jonathan Hardy were kind of the big players in a lot of the research that you saw out there. So thank you very much for your time, and I'll take any questions you guys have.